You may have heard about DNA paternity testing. In today's video, we'll talk about the genetics behind paternity testing and how it is used to confirm who the father of a child is. To find out more, keep watching.
Welcome to Ask a Geneticist with Mkwoto Pius.
DNA paternity testing is used to find out if a man is the true biological father of a child. It can be ordered for several reasons. It can be ordered by the parent who wants to find out if they are the real biological father of that child. It can also be ordered by the court to determine the paternity of a child for reasons of immigration, child support, etc. But it can also be ordered to find out the risk of a child having a genetic disease that is inherited from the father. So how is DNA paternity testing conducted? To conduct DNA paternity testing, you need two important samples. You need one sample from the child in question and a second from the presumed or assumed father of that child. These samples are mostly chick swabs, what we call buccal swabs, and it involves using a cotton swab to swab inside the chick to take chick cells. But sometimes blood tests can also be used to conduct DNA testing. Now, how does DNA testing work? For every child that is born, the father contributes half the genetic material and the mother contributes the other half of the genetic material to make the child. What this then means is that if you take the child's genetic material and the genetic material of the assumed father, you should find similarities between both of them. And this is how paternity is determined. What happens is that the samples are taken to the lab and put through a process called DNA sequencing. During DNA sequencing, we sequence the DNA for both individuals to find areas of similarity between both of them. If you've enjoyed listening to this, then subscribe to my YouTube channel and follow my Facebook page, and I will see you next week with more stories from our DNA.